Oh, that's so annoying. And with that, I would like to introduce Calvin Bond, who will introduce our speaker for tonight. Calvin?
Thank you, Ron. It is my pleasure to introduce Dr. Stepfanie Aguillon, who grew up in southeastern Arizona and earned her BS and MS degrees from the University of Arizona, where she began her career studying Western bluebirds. She later pursued her PhD at Cornell University in the Cornell Lab of Ornithology in upstate New York, focusing on the study of northern flickers. Recently, Dr. Aguillon joined UCLA to establish her own lab here in Los Angeles. As an evolutionary biologist, Dr. Aguillon investigates the origins and mechanisms of reproductive isolation in nature. Her research uses genomic sequencing, behavioral ecology, and museum science to understand the complexities of evolution, especially in species that naturally hybridize. She is particularly driven to understand how reproductive barriers and phenotypic differences emerge and how individual behaviors can shape genetic patterns across landscapes. Through much of her research, Though much of her research addresses basic science questions, Dr. Aguillon also applies these methods to conservation, contributing to population genetic studies and conservation unit definitions for species like the Bay Area song sparrow and the endangered Florida scrub jay. Her work bridges basic science with conservation, enhancing our understanding of evolutionary forces that shape biodiversity. Please join me in welcoming Dr. Stepfanie Aguillon.
Well, thank you. Excited to be here.
All
Let
right.
me share. And
Perfect.
should be able to see the slides now. Great.
It looks
Um,
great.
Wonderful. Well, I'm really excited to be here. I'm also really excited that you all are going to hear from Allison Schultz soon. Allison is a good friend and colleague, and you'll see some, I think maybe one or two mentions of her work here in this presentation. So I'm going to tell you a little bit about the work that I do, but I thought I would start by telling you a little bit about my academic journey and sort of how I got to be where I am now. Um, and so I often do that with a map, just because I think that adds a little bit of I don't know, helps people understand a little bit more about where I'm coming from. So I grew up in Sierra Vista, Arizona, which, if you are a birder and from the Southwest, you might know from the Sky Islands and places like Ramsey Canyon and the Huachucas. So that's where I grew up, which is a really incredibly biodiverse area and a really rich and wonderful place to be. And it really was a wonderful place to learn to be a biologist. So I, for instance, got to see many birds that are not really visible anywhere else, many mammals that are not really visible anywhere else, lots of herps and reptiles. I'm realizing maybe you can see this bar in the back here, so I might move it for you. Nope, it doesn't let me. Hold on. Want to make sure you can see the pictures well.
Now we see your sidebar.
Wonderful.
Okay, now we don't.
Great. Perfect. Um, so it was this really wonderful place to grow up, and lots of biodiversity, lots of animals. And this was really great for a kid like me who was really interested in being outdoors. So I spent a lot of my time hiking and camping and fishing, and really learned to love being outside. So it was not a surprise that I went to college to be a biologist. So I went to the University of Arizona where I got my bachelor's and my master's. And at this point, before college, I had not really known birds as something that was particularly interesting or exciting. I mostly spent time fishing or camping. But I discovered birds as an undergrad, actually. I took ornithology as a junior, and I was just enamored with birds and basically have not looked back since. So from the University of Arizona, I went on to upstate New York. I did my PhD at Cornell, where I learned a lot about flickers, which is what I focused on for my PhD, but also learned a lot about cold weather and what it takes to live in a cold place. So things like walking home when the buses are canceled because it's a snowstorm. Um, and sort of unsurprisingly, I moved back to California and the West Coast. But in between that, I got a lot of time and opportunities to do many wonderful trips for conferences, for teaching. So I taught a class that went to the Galapagos twice, which was really wonderful. And yeah, I visited many wonderful places just through the opportunities I got as a graduate student. Um, and that really has shaped a lot of what I do career-wise. Um, but I have since moved to California. I spent some time in the Bay Area at Stanford. After my PhD, I was actually studying fish using a lot of the techniques I learned in birds. And then I have recently moved to UCLA. I've started my own lab. This is me with my two grad students and postdoc. I started actually July 1st of last year, and so I've been in LA for about a year and a week or so. Um, and yeah, it's been really wonderful being at UCLA. And I'm really excited to tell you a little bit about some of the things that I'm really interested in research-wise and some of the directions we're, moving in for the future. So my research and sort of at the core of my interests is coloration, and it sounds like you'll hear a lot about this too in the next webinar with Allison. But I really am interested and have been fascinated by color in nature and sort of personally. I do a lot of handcrafts and embroidery that uses color and color theory. And this is a fascination that has not, is not just me as a biologist, but something that has fascinated biologists for a really long time. So this is an illustration of Darwin on the Beagle looking at a butterfly. And in a time where, there were no cameras and it was really like you trying to describe something to other people. You did this with what are called color swatches and Werner's Nomenclature of Color, which is a book that had swatches, and you could look at a thing in nature and say, it's this color blue, and other people would understand what that means. Of course, I don't study color in exactly this way anymore, but this, fascination with coloration in nature is, a longstanding thing in biology. For me, in addition to coloration, I'm also really interested in hybridization. And I'll, get to how these two interact. Um, but this is just to introduce hybridization. Probably you've heard about horses and donkeys being mated to produce mules. If you know about this example, you might know that mules are really hardy pack animals, and they're great if you're, trying to go down into the Grand Canyon, but they can't produce any offspring themselves. So you're not really mating mules to get another second generation of mule. And so you might think from this, they're like sort of a dead end. They're not really an interesting biological phenomenon. And I wouldn't be surprised if you thought that, because for instance, if you search hybrid animal online, you get photos and images that look like this. Obviously, these are not real. Um, and so it wouldn't surprise me if you thought hybrid animals are not that interesting or exciting. But actually, hybridization is not always an evolutionary dead end. So these are examples here of a number of different organisms from different taxa that have had hybridization in their evolutionary history. This is ranging from things from yeast to plants to humans. So we even have hybridization in our evolutionary past. This shapes things from, susceptibility to COVID, to lots of other things. And so hybridization influences a lot of things. And in birds in particular, hybridization is actually extremely common. So here's a bunch of examples of either hybrid individuals or species that have hybridization in their group. And hybridization intersects with my interest in color in some interesting ways, because many situations where we have hybridization in birds, we know about them because of the way the bird looks, because of its color, because of its patterning. And so we can really highlight this with one of these warblers. So this warbler is produced by hybridization between a blue-winged and a golden-winged warbler. It produces this hybrid Lawrence's warbler. And hopefully you can see from this Lawrence's warbler, it gets its overall yellow coloration from the blue-winged parent, but it has this black mask and this black bib from the golden-winged parent. And hopefully it's not too much of a stretch to say that this is due to underlying genetic differences. So the genomes of these two birds mix, and then you end up with coloration that is mixed. And so for this story, and for what I'm going to tell you about tonight, I'm going to take you to the Great Plains. And that's because not only are there bison now, But there are a bunch of hybrid zones in the Great Plains. This is what's called a suture zone. This is a geographic region where different biomes sort of come into contact. And so in this case, we have these east and west biomes. And there's hybrid zones in things like indigo and lazuli buntings, spotted and eastern towhees, black-headed and rose-breasted grosbeaks, bullocks and Baltimore orioles, as well as a bunch of other bird species, a bunch of plants, a bunch of mammals. Um, I'm not going to tell you about any of these ones on the slide today. What I'm going to tell you a lot about is flickers, which also meet in the Great Plains. So flickers come in what I like to call two flavors, red shafted and yellow shafted. And if you know anything about flickers, you'll know that they are really broadly distributed. So basically across all of North America, you can see flickers. If you're in the West, so in SoCal, you'll see red shafted flickers. Um, in the East, you'll see yellow shafted flickers, and I'm actually in Indiana right now, so we have yellow shafted flickers here. That was one of my goals for this trip I'm on this week to see a yellow shafted since I've been away from the East for a while. But in this broad region in orange in the center of North America is a hybrid zone. And this is where red shafted and yellow shafted ranges overlap, and you get lots of different hybrid individuals. And we actually know that hybridization has been happening for quite a long time. So as far back as Audubon, we have written record of flickers. So in the early 1800s, during Audubon's collection across North America, he wrote about a hybrid nest of flickers that they're one of the most curious set of 5 birds that he had ever seen, and that he thought would puzzle all of naturalists in the world. And this is one of the slides I would put up a lot during my PhD to just be like, wow, this was something that was supposed to puzzle all naturalists in the world, and I decided to study this for my PhD. Um, so hopefully I'll tell you a little bit more about what we've uncovered. But this is just to say that we know that hybridization has been happening for a long time. It's been in the written record for a long time, and it has likely to have been ongoing for centuries. And the reason Audubon was able to identify that hybridization was happening is that the flickers differ in a bunch of different plumage patches. So I think, very clearly from these illustrations and from seeing these birds in nature, you'll probably know that they differ in the color of the wing and the tail. This is what's called the shaft. It's either this salmony pink or salmony red color in red shafted flickers, or this really bright, vibrant yellow in yellow shafted. But if you zoom in on the head and face, sort of in like a NFL mascot style, head to head, you'll see a number of differences there as well. So males have this malar stripe, this mustache stripe that's either red or black. They differ in the throat color, gray or tan, and a number of traits on the top of the face and head as well. So the ear coverts, the crown color, and then yellow shafted flickers have this really broad red patch on the nape of the neck, the nuchal patch, which is absent in red shafted flickers. So these two, red shafted and yellow shafted, differ in a bunch of these traits. And hybrids end up with a variety of combinations of these different traits, as well as intermediate colors. So you can have an individual with orange wings, you can have an individual with red orange, yellow wings, and all other red shafted traits. So it's really easy when you're in the hybrid zone to see individuals that are really mixed and matched in phenotype. So that's a little bit about flickers. I'm going to tell you now 3 sort of specific science stories about them. The first one is looking at plumage coloration across the hybrid zone. Next, I'm going to get into genetics a little bit and tell you a little bit about genetics looking across North America, across different flicker populations. And then we'll really dig into genetics in plumage color in particular. And so we'll start with this, thinking about the hybrid zone. So as I mentioned, the hybrid zone has a long history of study. So this is something that scientists have been thinking about for a long time. We know from collections that were made in the mid 1950s that as you span from the western part of this hybrid zone over to the east, you go from red shafted like traits through intermediates into yellow shafted like traits. And one of the reasons that this collection is so exciting is that it lived in the museum where I did my PhD at Cornell. So we had these specimens from the 1950s, and I'm sure as Allison will tell you a lot about, um, museum collections and birds retain color if you care for them in the right way and maintain your specimens. You can have specimens from the 1800s that look just as good as a specimen from today. So we know from the 1950s how this transect as you go from east to west looks. And so I was really interested when I started my PhD in understanding how the hybrid zone had changed from the 1950s. It had been 60 years, and I was curious to know what was going on there. And so this was really a perfect opportunity to sample across this same transect. So if you think about the Great Plains and especially what flickers like woodpeckers need are big trees. The Great Plains doesn't have a lot of big trees. They're mostly along river corridors. And this collection in the 1950s was along the Platte River. And so it was a great opportunity to basically go back to the Platte River and collect at the same localities. And so this is what we did. Um, we ended up, um, as a result of this, with 250 samples from the historic sample from the 1950s. And then when we went back out, we did a contemporary sampling of a little over 100 individuals. So we have a pretty good comparison between these two time points. And this was all work in collaboration with the curator of birds and mammals at the CUMV, Vanya Rower. So I want to highlight him here. He's a co-author on all of this work, but I think he also deserves recognition in the talks. Um, and so what we did for this project in particular was described plumage color using a hybrid index. And basically what this means is we scored a bunch of these traits that I described to you, and we scaled it to range from 0 to 1. A 0 score indicates a pure yellow-shafted individual, a 1 score indicates a pure red-shafted individual, and values in between indicate different levels of intermediate plumage. And so when we look at these repeated transects, we can get these really interesting lines. And I'm going to tell you a little bit about how to read a cline. So this is what a geographic line looks like. You have distance along the x-axis and some sort of phenotypic trait on the y-axis. In this case, we can think about the color of the wing and the tail. On these clines, we have values for one species on one end of the cline, values for another species on the other end of the cline. We have this S shape where in the middle here is the hybrid zone. We have intermediate values for this phenotypic trait. So this would be where we have orange individuals for wing and tail. We can do some really interesting things with these clines. What I'm going to highlight here is just the ability to look at the cline center. So we might find that if we sample this cline historically and go back to sample it in contemporary time, we might see that the cline is in exactly the same place geographically. So that the center where this S goes down from the top to the bottom hasn't moved at all in geographic space. Alternatively, we might go back after our historic sampling and sample again and see that the cline has moved either to the left or to the right on our transect. This could be, north, south, east, west, depending on how your cline is oriented. But we could have some amount of movement here just based on the way these clines look. So these are the kind of results that I will show you here. So when we look at the flicker hybrid zone, we see that it has actually shifted quite significantly westward. So if we look at what this cline looks like historically, we go again from red shafted like to yellow shafted like as we grade from west to east across this cline. This is what the historic cline looks like. When we look at the contemporary cline, we see that it's moved left on this plot along distance. In this case, this means west. And this is true for individual traits as well. So this plot on the left is the overall hybrid index. But if we look at individual traits, we see that those have also shifted westward. So overall hybrid index, that's just the summary from the left. But individual traits too have moved westward, it seems, as well, including the wing and the tail color of the shaft. If we look at what this looks like on the map, so this is our sampling scheme. In the 1950s, the hybrid zone was centered at this point, sort of near the junction between Nebraska and Colorado, that corner. And in the 2010s, this has shifted significantly west towards the Front Range. So point 1 is basically getting really into the mountains. So it's moved quite significantly. So circling back to this question, so how has the hybrid zone changed over the last 60 years? We have this significant westward movement, and this is towards the range of the red-shafted flicker. And, why might this be happening is, I think, an important question. Um, so we know this has probably happened rapidly over the past 30 to 40 years. Previous work in the 1980s suggested that the hybrid zone had not moved at all. This was not using a similar, a repeated transect, but was using museum samples as well from this region. So we think this has happened since the 1980s. This may be the result of climate change. We know, for instance, the Oriole hybrid zone and the Bunting hybrid zone have also shifted westward. Um, so it could be climatic influences affecting all of these. It could also be differences in land management. So the way that the Platte River and the Great Plains have been managed has really changed with increase of white settlers basically into the Great Plains. These birds, flickers, and other hybrid zones in this area require big trees. And we have both planted those in yards in the Great Plains, but also the way we have managed the Platte has led to more big trees. So it's possible that you have more big trees in the Great Plains, and it's easier to flow into the Great Plains from the east than it is from the west. And so as a result, the hybrid zone is moving westward. We don't really know why this is, but this is something that we're actively working on. So maybe we will know soon as my lab continues to do work. So that's a little bit about plumage coloration in the hybrid zones. We know the hybrid zone is moving. But what about genetics in this system? So as I brought up at the beginning, flickers differ in a bunch of different traits. They have the wing and the tail differences, a bunch of differences in the head and the face. And so people have been really interested in these birds for a really long time because they seemed like a good system where you would find a lot of genetic differences. But surprisingly, all of the previous genetic work using a variety of techniques from allozymes to mitochondrial DNA to early genomic approaches could not find any differences either in the hybrid zone or across the species range. So when I started my PhD, I was really interested in understanding if modern genomic sequencing technologies could help us identify genomic differences between these flickers. The fact that they differ so greatly in plumage suggests that they, should have genetic differences too. So I'm going to show you methods for this just on one slide, and I'm happy to answer questions, but I feel like it's more interesting to talk about the science here. So what this work requires is some kind of blood or tissue sample, surprisingly, a lot of molecular lab work, and then more surprisingly, a whole lot of computer work. So actually, I spend most of my time on the computer work, even though what got me interested in the direction I've taken is the field work. Um, and so I wanted to, for this project, really maximize the potential to identify any genetic differences. So I took this approach where I sampled flickers really far away from the hybrid zone. So as far away as I could go in the west and as far away as I could go in the east, taking advantage of the fact that these birds are in many museum collections, um, and really trying to see if, getting as far away from hybridization potential as possible to see if that could help identify genetic differences. So the approach I took with this project is called double digest restriction site associated DNA sequencing. I'm not going to test you on that, but just to say this is an approach that basically takes the DNA and cuts it. So it's looking for these it's looking for these restriction sites, and an enzyme goes in and chops the DNA into these little bits. And then these small chunks are what is sequenced. And within these chunks, we're looking for what I'm going to call markers here. Um, and we can think about markers like this. So if we sequence three individuals, we see in their genome the different base pairs, the T's and C's and G's and A's. We can look within them to see what is the same and what is different. So in this example, we have two yellow shafted and one red shafted. We can look in their genomes and ask what is different. In this case, it's this spot here where we have a G in the red shafted and C's in the yellow shafted. This would be a marker or a single nucleotide polymorphism. This is just a region in the genome that's different. With this approach, we end up with about 9,000 markers across the genome, so many more than was previously possible. And we can use these markers to understand how these birds are related in their genome. So when we do different types of clustering analyses, we find that we can distinguish these flickers, which is the first time we've been able to do that. Um, so if we look at what's called a principal components analysis, each point here is a different sample. All that really matters in these principal component analyses is that the different clusters, red individuals cluster with red, yellow clusters with yellow, they cluster separately. So we can see that they do cluster differently. We can do similar clustering analyses with different types of programs. This is what's called a structure or admixture analyses. In this case, each bar is a different individual, and the amount of color in that bar indicates their genomic ancestry. So you can see yellow shafted individuals have nearly all yellow in their bars, red shafted individuals have nearly all red. So we can cluster these individuals as different, but they're actually still really genetically similar. So if we look at each of the genetic markers, we can look at a measure called FST, which is a measure of genetic differentiation. It varies from 0 to 1. 0 indicates that region in the genome is completely identical. 1 indicates that region is completely different. And so if we look at these 9,000 markers across the genome, we see a distribution that looks like this. The average is about 0.024. So using these 9,000 markers, we see pretty low FST, so pretty genetically similar. And even more interesting is this region of high FST from 0.75 to 1. We see very few markers. So there's only 7 out of these about 9,000 that reach this sort of high FST difference. And we don't see any markers that are at this 1 point. So there's no markers in this data set that are completely different. So we couldn't take one marker and say, this could tell us if we have a red shafted or a yellow shafted. So this is very interesting. So using these modern genomic technologies, what could we find out? So we were able to identify genetic differences. So we could use clustering analyses to say red versus yellow if we use the whole data set. But it turns out that they're really genetically similar nonetheless. This is probably why it has been historically so hard to find any genetic differences, because in the past we're using, 5 or 10 markers across the genome. In this case, using 9,000, we could only, really identify clusters here at this level. Okay, so that's a little bit about the genetics across different populations. I'll tell you now a little bit about plumage coloration and genetics. So flickers are a really great system for thinking about the genetic basis of color. So as I've highlighted a few times, they have these multiple distinct coloration differences, the wing and the tail, differences in the head and the face. They also have this extensive hybridization, and I will tell you a little bit about why that's important for looking at the genetic basis of color. And as I just mentioned, they have this low level of background genomic differentiation. So this means they're really genetically similar. So if we can find places in the genome that are really different, those might be related to color. So I highlighted at the beginning that hybrids have a variety of trait combinations and colors. So from, different mix and match of the different traits that they have, as well as, you know, intermediate colors like oranges in the wings and the tail. Just to show you this, is true in birds in the hand. This is not just true in these illustrations that I have. So to highlight that here. Um, and in addition to having mixed up colors in the plumage, hybrids also have mixed up genomes. So we can think about the genome here as these bars with different colors. They have, different combinations of red and yellow, and the color indicates what species that genomic ancestry comes from. And we can use these hybrids to try to get at the genetic basis of color. So if we're interested, for instance, in the red wing and tail, we can look in hybrids that have the red wing and tail and ask which region of the genome do they share that is not shared with other hybrids. So in this sort of toy example, this would be this region in red here at the top of this genome. So one of the things I was really interested in and continue to be interested in is what is the genetic basis of these coloration differences in flickers? So again, I'll show you methods in just a single slide. So this, in this case, I'm using whole genome sequencing. So this requires a blood or tissue sample, molecular lab work, and a bunch of computer work. In this case, this work ends up sampling across the entire genome. So I went from 9,000 markers in the previous part of this talk to over 7 million markers in this part. So we're really sampling a huge swath of the genome now. And I'll show you first what the results look like on a single chromosome and then show you the whole genome. So we can view these results in what's called a Manhattan plot. So the x-axis here is the position along the genome. In this case, we're looking at just a single chromosome. So we can see position along chromosome 10. And then the y-axis is this genetic differentiation measure, FST again. Remember that 0 indicates a marker is completely identical, 1 indicates a marker is completely different. And hopefully what you see in this figure is that as you move along this chromosome from left to right, you have things that are very, very similar, very, very similar until you get to the end, where we have this region that's really different. And these plots are often called Manhattan plots. And that's because you're sort of looking for these buildings that pop up against the city skyline. Um, and so we're looking for these differences, which are sort of represented as these buildings. And these are results with allopatric individuals. Allopatric means far away from the hybrid zone. So we're trying to get, as maximize the amount of genetic differentiation we might observe here. So this is just a single chromosome, but we can also look across the whole genome. And when we do that, we see low differentiation, but a number of these skyscrapers. So this is a Manhattan plot. You read this just the same as the last figure. This time though, the x-axis is different chromosomes. So you see, from chromosome 1 all the way to chromosome Z. This Z is the sex chromosome in birds. And as you can imagine, plotting over 7 million points is difficult. Actually, the average here is very, very low. So there's a lot of points here at the very bottom of this plot. The average across this genome is 0.008. So these birds are nearly genetically identical. But there are still a number of these peaks, so a number of places that they're different. Despite that difference, these birds are really similar. So out of 7 million markers, only about 900 or so are fixed differences, so points that reach this FST value of 1. So these birds are really, really similar. And so these are birds far away from the hybrid zone, but hopefully you remember that I mentioned there's a lot of potential in the hybrids. And so we can use these hybrids to do an analysis to try to understand if particular genomic regions are, associated with particular color phenotypes. And so when we do that, we actually find a number of interesting regions of the genome. And I'll highlight here just the analysis for the wing and the tail. So when we do this analysis for this color phenotype, we get a Manhattan plot that looks like this. The only difference from this Manhattan plot for the previous is that the y-axis here represents the strength of the association between a particular genomic region and a particular feather color. So in this case, you see a number of these peaks here, a number of these skyscrapers. These are regions of the genome that are strongly associated with the wing and the tail color and that particular place in the genome. We've done this as well for all of the different color traits. I'm going to only highlight the wing and the tail here and then instead move to talking about genes in particular. So within these regions of the genome, there are actually many genes that are known to be related to pigmentation. So a number of genes related to melanins. Melanins produce blacks and the browns and the grays that we see in nature. So a number of genes there. A number of genes related to carotenoids. Carotenoids are what produce the bright reds and bright oranges and bright yellows. A number of genes there as well. Some of these genes have really clear hypothesized roles in flickers. So CYP2J19, for instance, is one of the only genes we know in birds that changes yellow feathers into red feathers. And we find this related to the wing and the tail differences in flickers. So we think CYP2J19 is working with these carotenoids. Carotenoids have to be ingested. And so changes the ingested carotenoids in red shafted flickers from a yellow color to a red color. But we actually have a lot of melanin genes. And one thing that was really intriguing to me was when I started to try to look into what traits these genes were associated with. And interestingly, they were often carotenoid traits. So things like the malar stripe, this stripe on the face of the males, the nuchal patch, this big red patch on the nape of the neck, even the wing and the tail color. And melanins and carotenoids are not known to be influenced by the same genes or be produced by the same biological processes. Melanins organisms can produce in the body, carotenoids have to be ingested. But there are very different biological processes to produce these different colors. And so we're really interested in trying to understand why we're finding carotenoid genes or melanin genes related to carotenoid traits. I don't have an answer for why this is, but I'll walk you through a little bit about what's going on in flickers. So feathers are really complicated structures. Going from the pigment present in a feather out to the color that we see, there are a lot of steps in between. You'll notice that this is a slide from Allison Schultz. So I'm sure you'll see this again in the next webinar as well. But genetics are likely involved in many different steps of this process. And so it's potentially that there's a more complicated relationship between just like this gene we know influences melanins and the final color that we see. And aberrant flickers maybe can provide a clue. This is something that Allison and I have been really interested in. We wrote a piece on community science and aberrant birds in general that I don't have slides for actually, but I'm happy to point people to. But so here are some aberrant flickers. We have two yellow shafted and then a red shafted on the right. And I'll highlight this bird here in the middle. This is the individual that got me interested in aberrant birds during my PhD. So it was yellow shafted flicker that was sighted in Massachusetts in 2015. And I'll put here what yellow shafted flickers usually look like on the right and what red shafted flickers usually look like on the left. I think you can see from this aberrant individual, its body is just flush with carotenoids. So it's yellow all the way across its body. So it potentially isn't the genes here are maybe not influencing where carotenoids are deposited, since it looks like they sort of just put them everywhere in the body. Um, but I want to highlight in particular the malar stripe, this stripe here on the head and the face, which in typical yellow shafted individuals is black. But hopefully what you can see here is that this individual has a red malar stripe, which is like what's seen in the red shafted individual. So it turns out what yellow shafted individuals typically do is take this yellow patch that is already present and just put melanins on top of it, producing a black patch. So when you take feathers from the malar stripe of a yellow shafted individual and pull out the melanins or try to chemically identify melanins and carotenoids, you find both of them present in these feathers. So perhaps it's less about what's happening with the carotenoid genes themselves, but what's happening with the melanin genes and where melanin is being deposited on the body is really influencing the color here. So this is something that we're really actively pursuing and interested in, and hopefully we'll have more answers in a few years. But yeah, this really interesting complex relationship between what color a feather is and what pigments are present in it. Okay, so what's going on with the genetic basis of color in flickers? So I showed you that there are a number of differentiated regions in the genome. These are associated with coloration differences. Overall, these birds are really, really genetically similar. But within these regions that are different, we find these complicated but interesting patterns with different genes that are present there that influence pigmentation. So I told you today a lot about flickers. Hopefully you can both identify these birds and appreciate them a little bit more. And in terms of what I told you about the science, so the hybrid zone has moved west in the last 60 years. we are really interested in seeing what this means in the future if the hybrid zone continues to move. I have strong suspicions that it will never get past the Rocky Mountains, um, but we will see. Um, I've told you a little bit about looking broadly at birds across North America and looking at their genomic differences. We are able to differentiate them based on modern genomic sequencing technologies, but they are still really genetically similar. And then I told you a little bit about my work on coloration. And again, when we look at the whole genome of these birds, we find really few regions that are different between them, but the majority of them are related to coloration differences, and contain many pigmentation genes, some of them like CYP2G19, with pretty clear hypothesized roles. And hopefully we'll know more about exactly what those are doing in the future. Okay, so with that, happy to take any questions. Um,
Fantastic. Thank you very much, Stephanie. That was
of
absolutely
course.
wonderful.
Yeah, fantastic. Thank you so much.
Anyone
For
with if you have questions for Stephanie, please pop them into the Q&A section down at the bottom of your screen. That helps us keep track of everything.
Yeah. So I already have a question that Janet has forwarded from the live stream. So Steve asks, I live on the Olympic Peninsula in Northwest Washington. We regularly see red-shafted flickers in the breeding season with red nuchal stripes. There's a juvenile with one in my backyard today. Thoughts?
Yes, that is a great question. Let me see if I can get here back to the map just to highlight the region we're talking about. A bigger map. This one will do. Yes, so this map here is a bit stylized. So the orange region here in the center, and maybe if I do like this so I can highlight here. So we have this region here in orange goes all the way up. So the hybrid zone goes from like northern Texas up through the Rockies into Alaska.
Yep.
And then this region here is much broader in the hybrid zone. So, and particularly around the nuchal patch. So, in the Pacific Northwest, it's pretty common to see red-shafted individuals with some amount of red in the nape of the neck. It's unclear exactly why. I don't know if that's a trait that has sort of introgressed or moved with hybridization into that part of the range, or if the hybrid zone is just still really present there, and we're only mostly seeing that in the nuchal patch. We do see birds, this is true in Southern California as well. We see birds that are some amount of hybrid um, that are present often in the winter, typically in Southern California. And I think what's happening there is birds sort of from the hybrid zone in Canada migrate down and overwinter like in California. That's something we're really interested in. I actually don't have it in this presentation, but we started putting GPS trackers on birds in California, and have found that one of the birds we tagged in the Bay Area actually went north into Canada and basically crossed the Rockies in Alberta, and was breeding sort of like all the way on the other side here of the hybrid zone, and then came back the next winter to the same location in the bay that we tagged it.
Wow.
So I think there is a complicated relationship, especially in the West. Um, and we don't exactly know what's happening. But that's one of the things I would be interested in understanding more. So hopefully we'll have more genetic samples and more GPS tags in the future to sort of know where individuals are going and where they're coming from.
Wow. Wow.
Wow. Oh, fantastic. No, that's really interesting. I have a similar question from Pete. Why would different traits move different distances? Is there anything known
Yeah.
about mate selection preferences and color traits?
Yeah, that's a great question. So surprisingly, and I think this is one of the reasons I was really interested in these birds, is that they don't seem to care about the color of the individual they're choosing to mate with, which is not really what you would expect, based on the fact that the hybrid zone has been maintained in this region for so long. You'd actually expect that they would, like, red shafted would mate with red shafted, yellow shafted would mate with yellow shafted. Um, and so it's unclear why different traits are moving at different rates. It's possible that it's a sampling issue, that we have just not sampled enough individuals, and the ones we have sampled have had, you know,
Hmm.
a unique combination of traits that makes things look like they've moved differently when they haven't. Alternatively, it could be that it's related to the genetic basis of these traits. So I'm really interested in understanding if some of these genes that underlie these color traits are working together, or sort of inhibit each other. So, like, if you have one gene to produce red shafted wings, like, it's harder if you have another gene that is related to, the nuchal patch, that causes them to work against each other, which could influence the way these are moving across the hybrid zone. But we don't really know yet. We are increasing sampling and doing more whole genomes, and also getting more into really what's going on structurally in the genomes of these birds. And so I think we should have a better handle on that pretty soon.
Excellent, excellent.
Thank you. John, well, it's not really a question, but he says, John says, excellent presentation with fantastic use of graphics that really helped clarify everything. Good work.
Thank you.
So
A
fantastic.
very kind
And
comment.
I fully agree. Oh, I see.
Absolutely.
Tori, do you have a question?
I do. Thank you. This is fascinating, really, really interesting. One of the questions that I have, and I don't know if you know about this, but you're probably much more up on the literature than I am, which is that I know people are beginning, now that we have genetics being such a presence in the labs and helping our understanding of species and hybrid zones, we're seeing hybrid zones shift in a lot of different ways. This is one example of one. I know there are other cases. If we look at the titmice, we look at situations where hybrid zones are getting wider or narrower, or moving north or moving west. And I'm wondering if there's any consensus amongst the communities of people who are looking at hybrid zones to try and make sense of these shifts in a larger context. Like, are there patterns? Like, you'd expect a north-south movement, maybe because of climate change, but some of these other ones are a little bit harder to figure, including like changing the breadth of the hybrid zone is really interesting. If you have anything to share on that. Thanks.
Sure.
Yeah, that's a great question. And yeah, I think there are some clear, like, as you said, like, hybrid zones moving north. I think we think a lot of those are due to climate change, with temperatures getting hotter. Things are moving to sort of track that, and that's changing where these hybrid zones are present. Um, Things like the chickadees in the east are a good example of that. Um, but there are a lot of other hybrid zones that are sort of more mosaic-y, like the golden wing blue winged hybrid zone is sort of patchily distributed.
Hmm.
Um, but yeah, I think these ones in the Great Plains that are, sort of north-south oriented, um, the the hypotheses for why we might be seeing a westward movement of multiple of these hybrid zones is a bit confusing. So I wouldn't say there is a clear consensus at this moment of, like, why that is. I think most people are sort of like, probably some sort of climatic changes are at play. Um, but we don't know for sure. And I will say, like, that's one of the things I had put in the paper about that hybrid zone movement, and the reviewers were like, "We don't know enough yet. Like, you can't say that." So, like, I think there isn't a consensus yet about why those are moving west. Um, I think in general there is, strong support that, like, climate is involved in hybrid zone movements, and so we know that from the sort of northward shift. But I think depending on the orientation of the hybrid zone and where exactly it is and what exactly type of climatic changes are happening in that region, I think it seems sort of idiosyncratic what's going on in each one. And yeah, I don't think there's a general rule yet.
Interesting, interesting.
I actually have a question too, Stephanie. I was wondering if you've ever done any work with like gilded flickers or like Guatemalan flickers or something where it's like a different
Yes.
species, but they're like they sort of have like gilded flickers. I know I don't know that much about the others, but it's sort of like intermediate traits, but it's like not a hybrid. Have you ever done like any work looking at those?
Yes. Yeah. So I did actually, yes. So the second project I talk about actually includes gilded flickers, but I omitted them for this talk because it's often easier to do so. Um, Gilded flickers are actually present in so this little part of North America where there is no color is gilded flickers. And I love gilded flickers because they're Sonoran desert specialists, and that's, you know, my backyard when I grew up. So I care a lot about them. And they're really interesting because they are basically look like red shafted flickers in the head and the face, but they have the yellow wings and tails of yellow shafted flickers. So there's really interesting combination of these two. And they're a little more genetically different from red shafted and yellow shafted. So you can sort of genetically differentiate them a little bit. Um, but we don't actually know what causes, you know, if it's the same genetic regions that produce the yellow wings and tails. We don't know if it's, you know, they first shared an ancestor with red shafted flicker and then yellow shafted flicker came next. So it's a little confusing. And I think that's all a little more muddied by the fact that gilded flickers hybridize with red shafted flickers as well, in sort of more of a like habitat dependent hybridization. So in regions that are like river corridors in Arizona in particular, you'll get hybrids of these two. Um, and it's hard to identify a hybrid when you know, they basically have the same head and face traits and they only differ in the color of the wing and the tail. Um, that would be something I would love to get more into. There's some work historically, but has not been a ton of work on gilded flickers since then. But yeah, I'm very interested in them. So yes,
Excellent.
Thank you. Yeah, that's so
yeah.
cool.
So I have another question. This is touching on something that you said about the flickers don't really care mating preferences with what color they are, and something that Tori touched on is the width of the hybrid
Hmm.
zone. So has the flicker hybrid zone been getting wider or narrower? And what would you expect if they don't care about who they mate with, that it would, you know,
Yeah.
continually get wider?
Yes,
And what
that's
do you see?
exactly right. That is exactly right. You would expect that it would get wider if they didn't care. If this balance between, you know, parental types coming in, and hybridization happening, and this sort of just like diffusing across the landscape, that would be basically the neutral expectation. And this was actually part of this paper, and I did not include that here because I felt like it would be too complicated. So I'm glad you asked it. But actually, the hybrid zone has remained just as narrow as it was in the 50s when we resampled
Hmm.
it. So it has remained narrow despite the fact that it has moved, and despite the fact that it doesn't seem like individuals care who they're mating with. And you know, the only reason you would expect a hybrid zone to stay narrow over time and not sort of flatten out is if there's some sort of selective process keeping the two types different on either end. And so one of the things I'm really interested in for my lab is trying to figure out what is keeping them different if they don't seem to care about the color of who they're mating with. And we don't really know yet. I'm sort of exploring the genomic angles and the migration angle. So flickers do migrate, and it's possible that if you are a hybrid and are migrating at some intermediate direction, that is very bad for you. And so we're trying to figure out some of those things. But at least for traits that people have looked at for birds just like in the field, there has been no clear like they don't seem to mate assortatively. Hybrids don't seem to do worse. They produce offspring just the same as everybody else. Um, so yeah, for those sorts of traits, there has not been like clear causal factor. So that's one of the things I'm really interested in trying to figure out with my lab is, you know, if that's true, why has the hybrid zone not widened. It's remained remarkably narrow.
Huh.
Interesting.
Very, very interesting. Something for the future and for new students to come in and
Yes,
take a look at.
exactly.
Absolutely. And I can't wait to find out on that GPS data that comes
Yeah.
in. That I'm looking forward to your next webinar, Stephanie.
Yes,
Yes,
absolutely.
yes, it's fantastic. You're also getting some compliments in the chat about with the thank yous and everything, which are
Oh, great.
Oh,
absolutely.
we have
Oh, we
another
have another
question.
question. Um, Lily asks, are the habitat preferences for hybrids like both of the subspecies?
Yeah, I mean, we think
Hmm.
so. That's so one of my grad students is going to do some modeling of habitat preferences. So I think we'll know a little bit more of that soon. But
Okay.
just, general what we know about the biology of the birds is they do basically the same thing. They prefer really similar habitat. They, don't seem to play different roles in their environment. They all eat
Hmm.
ants. We don't think they eat different ants, though nobody's really looked at that before. Um, and yeah, it's possible that the Great Plains serves as sort of like a sink. Like the habitat is not as good as what's on either end of it, and so hybrids are sort of maintained in that region because they're doing okay. Um, but we don't really know more than that. Like I think our suspicion is habitat preferences are pretty similar between the two types. Um, but I think we should know in more detail soon.
Okay. Great.
Great, thank
Mark,
you.
I think we're out of questions
We are at the moment, yes.
at the moment. Stephanie,
Okay.
thank you very much for a wonderful, wonderful webinar. Yes,
Yes, thank you
absolutely.
very much. And we hope that you'll come back and give us another webinar at some point in the future
Of
with
course, very happy
updates
to.
about this or anything else you'd want to talk about.
Absolutely, absolutely. And thank you very much for staying up. I realize it's kind of late over
Oh yes,
in.
it's a
It's
little bit later in Indiana than
it's very
yeah.
I'm very happy to do it.
Well, thank you very, very, very much. And please join us for our next webinar on the second Tuesday of the month on September 9th with, uh, God, her name disappeared out of my
Allison.
mind. Allison Schultz, yes. Which will also be very related to what Stephanie presented tonight. And so
Mhm.
we're really looking forward to that. And with that, anything else to add? I guess not.
Nothing for me, just thank you, Stephanie. It was fantastic.
Thank you very much.
Thank
And
you.
I will see everyone.
Thank you.
We'll see
Thank
everyone
you
September
all.
9th. Thank you. Take care, everyone. Good night now.
Okay.